All right, so this is the cell. Talking about the cell. First thing first, a cell is the basic structural and functional unit of an organism. So it's a basic part that controls what we do and which makes up a person. So without a cell, you can't have a person at the end of it, or because it's a basic building block. You have got two types of cells: a prokaryotic cell and a eukaryotic cell. The difference between these two types of cell is organelles which have got a membrane. Prokaryotic cells do not have membrane bound organelles. An organelle is simply a metabolically active substance found in the cytoplasm, in the space inside the cell. But not all of them are going to be surrounded by a cell by a membrane. Prokaryotic cell, to be specific, won't have membrane bound organelles. The nucleus is supposed to have a membrane. Because it doesn't because prokaryotic cells can't have those, the nucleus is not present in the cell membrane. Eukaryotic cells, on the other hand, are going to have membrane bound organelles. Any organelle that is supposed to have a membrane, including those who don't have a membrane, they are going to be found in eukaryotic cells. And just to mention examples of non-membrane bound organelles, the very common example is a ribosome. A ribosome does not have a it doesn't have a membrane. So you expect a ribosome to be found in a prokaryotic cell. But all other organelles that we can mention, anyone to mention, okay, let's each mention at least one organelle that we know. Mm, let's make it interactive so that things will be able to stick. Any organelle? Mitochondria, thank you very much. Anyone else? Endoplasmic reticulum, thank you. Where we have got the smooth and the rough, thank you so much. Anyone else? Is that all? Okay, we have got proteosomes. Yes, you can go on, please. Someone wanted to say something? Okay. So, of all the organelles you can mention proteosomes, lysosomes, you can mention a lot and a lot. Most of them are going to be membrane bound except the ribosome, so meaning that the rest of them will be found in the eukaryotic cell. Now, a cell has got two parts. It has got a plasma membrane and the cytoplasm. Plasma membrane is that thing that surrounds the cell. And all those organelles which you are saying membrane bound organelles, they also actually have a plasma membrane. Now the cytoplasm has got is just that part that's around the nucleus. The entire space that is inside the plasma membrane. And it's going to be made up of two parts, the cytosol and organelles. The cytosol is the liquid part that is inside the cell. And then organelles are those compounds which are dissolved. Okay, so here is what you can see: the cell membrane outside, which is a phospholipid bilayer because it has got a phospholipid, and you have got actually two of them. You also have cytoskeletons, the Golgi apparatus, the peroxisomes, which are used to chop long fatty acids, breaking them down. Mitochondria for energy, the cytosol. So all these organisms we are mentioning, they're actually found in the cytosol. Okay, the cytosol and the organelles make up the cytoplasm. Let's start with the cell membrane. The cell membrane, at least take note that it is a lipid bilayer. It separates things which are inside the cell. We call them ICF, intracellular fluid, the fluid inside the cell, from ECF, extracellular fluid. The intracellular fluid is what we also call as the cytosol. Please also take note for some questions the diameter of the cell membrane 7.5 to 10 nanometers. So this is how the cell membrane looks like. You can see these blue things on top here. These are actually the heads of a phospholipid, and then the yellow ones you can see these are the tails, which are the fatty acids. So you have got the heads pointed to the outside of the cell and the heads pointed to the inside of the cell, and then the tails are in between. They are held together by what we call hydrophobic interactions. Now, the cell membrane is going to have different components as you can see. It has got a lipid component which is made up of the phospholipid itself, which is the bilayer, which is the majority. It also has proteins. So you can see we have got proteins which will move from one end of the cell membrane going all the way to the other end of the cell membrane. Such proteins are called transmembrane proteins, also known as integral proteins. And then you have got proteins which only touch one end of the cell. One end, we call that as a peripheral. So these are known as peripheral proteins. You also have got carbohydrates as part of the cell membrane. For example, you can see this one which is in green, a glycolipid. This is just a carbohydrate attached to a lipid. Are we good so far? Is there any question? Are we good? Be very free to ask. I know you have done this topic. Maybe you didn't understand during a lecture. It's a time for you to get to ask. What are the functions of the cell membrane? It's there for protection, for permeability to allow things to enter or leave the cell, to absorb some things, okay, to excrete some things, to remove things from the cell, for gaseous exchange, and also to maintain the shape and size of the cell. The cell membrane is going to have proteins making up like 55%, and then which is the majority. I had mentioned lipids, but the majority is proteins. Lipids is like 40%, and then carbohydrates 5%. There's something that is known as a fluid mosaic model. This just describes how the arrangement of components. A mosaic is just having different arrangements of different things. Okay, so we describe the cell membrane as a fluid mosaic because it is made up of different things which can float on the lipid. We have got proteins which can float in the lipid layer. Actually, these proteins that you can see here, they are not stationary. They are not at one point. They can actually move about on the cell membrane. That is what we call the fluid mosaic, proposed by S.J. Singer and Nicholson in 1972. So the carbohydrates part of the cell membrane will mostly be involved. They are actually negatively charged. Okay, so they are going to be involved mostly in forming receptors. What is a receptor? A receptor is a component of the cell membrane that holds the things, that is able to hold things. For example, if anything wants to enter the cell, it is going to bind to a receptor, or anything that does not belong to the cell enters the inside of the body, we are going to recognize that there is something because it has been held by a receptor. Okay, so that is that. And then the lipid component of the cell membrane, there is a phospholipid and there is cholesterol. These are the many components. And then what is the function of the lipid layer? The lipid layer is there to allow the exchange of things. Those things which are hydrophobic, which do not love water, can actually pass through the cell membrane without the assistance of anything whatsoever. Remember, as we talked about yesterday, things like oxygen, carbon dioxide, and alcohol. While things which are hydrophilic, which dissolves in water, They can't pass through the cell membrane, like glucose, urea, and electrolytes. There is a question, please, you can go on. Or maybe it's a mistake. Did anyone raise a hand? Okay. Please take note that oxygen, carbon dioxide, alcohols can actually just pass through the lipid. Why? Because these things are hydrophobic and the cell membrane is made up of lipids. Okay. This is how a phospholipid looks like. It has got a head, which is made up of a phosphate, it's negatively charged, and then the tail made up of fatty acids. One is a saturated fatty acid, it has only single bonds, the other one is unsaturated. It has got a double bond, and that double bond makes it to have a kink or a bend. There are different types of phospholipids which are found in the cell membrane, like phosphatidylcholine, phosphatidyl ethylamine, phosphatidylglycerol, phosphatidylserine, phosphatidyliniso. So these are the different forms of phospholipids which are in the cell membrane. One thing to note is that the phospholipids are amphipathic. They have got the head, which is hydrophilic, and the tail, which is hydrophobic. And the tails are more to the inside, right? We remember what we said, that the tails are held by hydrophobic bonds, are held together like that, while the heads of one part will be facing the outside of the cell, the other part will be facing the inside of the cell. This is what we are saying. You can see the heads here, they are facing the outside of the cell. Here they are facing the inside of the cell, and then the tails are in between. Cholesterol is also amphipathic. What cholesterol does is when it is cold, prevents the cell membrane from freezing. When it is so hot, prevents the cell membrane from melting. And then it also has got a protein layer. The protein layer, the cell membrane is going to have integral proteins, which we also call transmembrane proteins, and peripheral proteins. As we said, you can see these ones which are passing through the cell membrane. These are transmembrane, also known as integral. The ones on the surface, these are peripheral proteins. Integral proteins, these ones which pass from one end of the cell membrane to the other, mostly involved, we talked about yesterday, to allow things to pass from the end of one cell to the other end. That is for cell transport, or as carriers, or as channels to allow ions to pass through. Others will be used as hormones, enzymes, while the peripheral proteins are mostly going to be enzymes. Some, of course, they can be carriers, okay, and then others will be proteins of the cytoskeleton. We'll talk about it. Is there a question on the cell membrane before we go to the cytoplasm? Okay, since there's no question, let me ask you, what is the biggest component? What part makes up the biggest component of the cell membrane? Carbohydrates, lipids, proteins. Okay, good. Now, the cytoplasm is going to be divided into the cytosol and organelles. Cytosol is the fluid matrix where organelles are suspended. Organelles, you can have those which have got a membrane, those without a membrane. Those with a membrane, mitochondria, nucleus, endoplasmic reticulum, Golgi, lysosomes, peroxisomes, vesicles. Non-membrane bounded cytoskeletons, microvilli, centrioles, cilia, flagella, ribosomes. So these don't have a membrane. So starting with now ribosome. The only thing to know about ribosomes, they are involved in protein synthesis. They are actually made up of two units. We have got what we call the large ribosomal subunits and the small ribosomal subunits. Okay. So these ones are going to be necessary in transcription to be able to convert DNA to, actually not transcription but translation, converting RNA to proteins. So these are ribosomes that we can see. You can have free ribosomes or ribosomes which are bound to the ER. Okay, so mRNA can be converted to proteins. mRNA can be converted to proteins. So this is the mRNA that you can see, and this is the ribosome. So when you have got a lot of ribosomes on the same mRNA molecule, we call that as the polyribosome. Polyribosome. And these ribosomes like this will be forming proteins which are destined to the mitochondria, to the peroxisome, and to the nucleus. Okay. And also proteins which are going to go to the cytosol and cytoskeleton. Any protein that is not folded well will be attached to what we call ubiquitin. If you just see a protein with ubiquitin, just know that that protein is supposed to be destroyed. And the organelle which is responsible for destroying proteins which have been coated with a ubiquitin is a proteasome. While those ribosomes attached to the mRNA, but then they're also attached to the rough endoplasmic reticulum, will be able to form proteins which are destined for the lysosome, proteins which are supposed to go to the cell membrane, and proteins that are supposed to go out of the cell. Just these three types of proteins: lysosome, cell membrane, and out of the cell. The rest of the proteins, which are supposed to go to the nucleus, peroxisome, mitochondria, cytoskeleton, cytosol, they'll come from free polyribosomes. The endoplasmic reticulum, this one is the largest membranous system of the cell, and it will make up like half of the total membrane volume. It's going to have a space inside it, we call it a cistern, and you have got two types of endoplasmic reticulum, the smooth and the rough. What determines if the endoplasmic reticulum is going to be smooth or rough? Anyone? Okay, all right. Thank you so much. The presence of ribosomes. That is correct. So, the rough endoplasmic reticulum, take note of this because it has got ribosomes, and you know that ribosomes are involved in protein synthesis. So, the rough endoplasmic reticulum is going to be involved in making proteins. And so, those cells in the body where the formation of proteins is more are going to have a lot of rough endoplasmic reticulum. For example, the pancreas. The pancreas makes up digestive enzymes, enzymes necessary for digestion. Okay, we call them pancreatic juices. So it's supposed to have a lot of rough ER because it makes up those proteins. Fibroblasts, we'll talk about connective tissue soon. Fibroblasts are cells which are found in connective tissue. They make up collagen, which is a very important component of connective tissue. So you expect them to have a lot of rough ER because collagen is a protein. Plasma cells, these are cells which produce antibodies. Antibodies are proteins, so expect them also to have a lot of rough endoplasmic reticulum. Okay. Other functions of the rough ER: production and secretion of proteins, and these proteins should not be destined for the cytosol. What makes up the proteins that are destined to for the cytosol? Anyone? What will make up, if we are saying that the rough ER will make up proteins but not those going to the cytosol, then what will make up proteins going to the cytosol? Remember we said that the free polyribosomes, or the ribosomes which are not attached to the rough ER, those are the ones making proteins which are destined for the cytosol. The rough ER is also involved in degradation of worn out organelles. Any organelle that has worn out, has been destroyed, the rough ER will be able to degrade them, to break them down. And then also the initial modification of proteins that you have formed, because proteins don't be, remember in first year we talked about translation. Proteins don't become functional unless you modify them, but that initial modification will happen in the rough ER. The smooth ER, on the other hand, the most important functions you need to know, they are called smooth because they don't have ribosomes. They are going to be involved in formation of glycogen and lipids, and also detoxification. What organ do you know that is involved in detoxification, removing of toxic substances? Very good. The liver. So you expect that smooth endoplasmic reticulum should be high in the liver. It is actually in the liver where we store glycogen, apart from the muscles also. Also, the lipid metabolism. It can either be anabolism or catabolism, breakdown or formation. And also temporary calcium secretion or sequestration. And in muscles, the smooth endoplasmic reticulum is specifically known as sarcoplasmic reticulum. It is involved in releasing calcium, and calcium is a very important ion for your muscles to contract. Without calcium, you can't contract. Okay, very important. So those are the principal functions. Detoxification, there is a family of cytochromes. They called cytochrome P450. These are the ones which are involved in detoxification, detoxifying alcohols, even drugs. Okay. And then we have got the Golgi apparatus or Golgi complex. The most important function every time you think of the Golgi is the processing, packaging, labeling, and delivery of proteins. Remember, the rough ER has made proteins, but then they are not functional. They are going to be processed, they are going to be packed, they are going to now be taken to where they're supposed to go by the Golgi apparatus. Okay. Also, some form of synthesis of carbohydrates will be able to happen in the Golgi. I didn't mention this thing, but one thing you need to know is look at this. This is the nucleus. Around the nucleus, we have got a rough ER. The membrane of the nucleus, because the nucleus is a membrane bound organelle, it has actually a double membrane, just like the cell membrane. The membrane, the outer membrane of the cell membrane is continuous with the membrane of the rough endoplasmic reticulum. So the rough endoplasmic reticulum always be near the nucleus. The Golgi apparatus will have one end which will be receiving proteins that have been made in the rough ER. And then one end, after those proteins go into the Golgi, they are processed. One end now will be able to release those proteins so that they go out now. Okay, so that end that will be able to receive those proteins, we call it the cis phase. And then the other end that is close, actually the trans phase. Okay, you can even look at here. Let me see if okay, I've got this nice diagram here. Okay, sorry, my bundles were done, so it had cut. Are you able to see the screen? Where did I end? I don't know where I'd... Have you gotten it? Did I reach on the Goji? Hello? Okay. So I was saying that the Goji apparatus is going to have the cis end. That is the end that will be able to receive proteins. Now, proteins, when they are made from the rough ER, they will be transported in what we call vesicles to the Goji. And then now it will be taken to come from the transphase and then go out of the cell or wherever they are sent. The mitochondria is going to make energy. That's a function. One important thing is that the mitochondria, the DNA found in the mitochondria comes from the mother. It's not inherited from the father. So if meaning there is any problem with the mitochondria, it means that the problem was actually from the mother's end. One other important function of the mitochondria is that it is involved in what we call apoptosis. What is apoptosis? Anyone? Programmed cell death. Thank you very much. It is programmed. For example, red blood cells, when they just reach, when they just reach 120 days, they're supposed to be destroyed. Okay? Supposed to be destroyed. So, when 120 days reach, the mitochondria will be able to destroy them. And what actually does that function is cytochrome C. Okay, cytochrome C involved in apoptosis. Also production of energy and synthesis of ATP. Peroxisomes, peroxisomes have got a single membrane. The mitochondria has got a double membrane, just like the nucleus and the cell membrane. Peroxisomes form what we call oxidases and catalases. Oxidases, these are enzymes which are involved in producing hydrogen peroxide. And then catalases, these are enzymes which are involved in breaking down of hydrogen peroxide. Hydrogen peroxide can damage the cell. So these enzymes, therefore, they do regulate like that. Okay. So peroxisomes are going to be involved in what we call beta oxidation. Beta oxidation is a process by which if you don't have enough energy in your body, you don't have carbohydrates, fats are going to be broken down so that they can give you energy. That is beta oxidation. Peroxisomes can be formed by two ways. They can bud off from the from the endoplasmic reticulum, or they can divide from already existing peroxisomes. They just divide on the middle. Lysosomes are also membrane bound organelles. They are formed by the Golgi. That is where they're going to be able to come from. And these lysosomes are actually phagocytic in nature. They are going to be able to take in anything that does not belong to our bodies and then be able to destroy it. That is phagocytic. They are phagocytes. So they are going to be found on cells like macrophages and neutrophils because these ones are phagocytic cells in our bodies. They are involved in phagocytosis. Okay, so one important thing about lysosomes is that they have got different enzymes which are involved in digesting things and breaking them down. And then proteasomes, these ones are going to be able to degrade denatured or non-functional polypeptides. That's the basic function of proteasomes. You also have what we call the cytoskeleton. The cytoskeleton, think of it, you have got a cell with the cell membrane and organelles. Organelles don't just float, they go this side, they go this side, they go this side in the cytosol. They don't do that. They are held together. And what holds them together is the cytoskeleton. The cytoskeleton is there to provide structural support and also for motility, movement of things in the cell. There are three components of the cytoskeleton: the microtubules, microfilaments, also known as actin filaments, and intermediate filaments. The biggest of these filaments are the microtubules, followed by the intermediate filaments and the macrofilaments. You can see microtubules, their diameter is 25 nanometers. Intermediate filaments, their diameter is 10. Microfilaments is from 5 to 7. Okay, mostly we say 7. Microtubules, they are going to be found also in what we call the centrioles, which are involved in cell division. You'll see them later. And also in cilia, flagella. Cilia is going to be found also in our nose. These are areas which are involved to push things. They are also found in the uterus, so that when the egg has been produced, it can actually be pushed from the ovaries all the way to the uterus. That's the cilia. Flagella is going to be found also like on things like the sperm, the tail for movement. Basal bodies, these are going to be able to anchor the cilia into the cell. For example, you have got hairs in our respiratory system, but they're going to be anchored to the cells so that they can actually stand upright by basal bodies. Microfilaments, these ones are just because they're made up of actin. Actin is important in movement, so they are involved in movement. We'll discuss more about actin and myosin when we talk about muscle contraction. So it's basically movement. Intermediate filaments, these ones also will be able to get to hold things within the cell like that so that they don't stop to move, or well, so that they are held closely in the cell membrane. We also have what we call inclusions. Now, inclusions are not organelles. Organelles are metabolically active substances. Inclusions are not metabolically active, and they don't have any membrane. Examples of inclusion, these are just things found in the cell membrane. They're not organelles. Fatty droplets, glycogen, lipofuscin, hemosiderin. These are just examples of uh, inclusions. The nucleus is going to have three main components: the nuclear pores, these are spaces; chromatin; the nucleolus. It's the biggest organelle in the body. You can see that it is continuous. The membrane, the outer membrane of the nucleus, because that's got two membranes, the inner membrane and the outer membrane. The outer membrane is going to be continuous with the rough endoplasmic reticulum. You can see inside the nucleus we have got chromatin. This is basically composed of DNA, and then you have got the middle part, which is the nucleolus, and then you have got pores. When your DNA is converted to RNA, that RNA has to move out of the nucleus to the cytoplasm using the nuclear pores. Now, the nucleus is going to have intermediate filaments. Remember, this is a cytoskeleton. Please, it's intermediate filament, and these intermediate filaments are known as laminins. Okay, and these will be able to bind membrane proteins and associate with chromatin in non-dividing cells. They'll just be there to bind proteins in the membrane. And then the inner membrane is going to have an organized meshwork, more like holding it together. And that is going to be called, what holds it together like that is a nuclear lamina. The nuclear lamina is just there to stabilize the nuclear envelope, that covering of the nucleus. Okay. And the major component of the nuclear lamina is the laminins. So this is what you can see here, holding the membrane together. It's more like a mesh, like a net. Now chromatin is just actually going to be non-dividing, is going to be found in non-dividing nuclei. Chromatin is going to be made up of DNA and proteins, which we call histones. Okay, and you are going to have two types of chromatin. You have got heterochromatin and euchromatin. Heterochromatin is a chromatin that is inactive, while euchromatin is a chromatin that is active. When you are, let's say, forming mRNA, you want to also form proteins. So how do you form the same chromatin and everything? You are first going to have one strand of DNA, another strand of DNA. When they coil together, they are going to now form DNA. That DNA will start wrapping around the histone proteins. That forms a nucleosome. A nucleosome will start to coil about itself. That forms now what we call a chromatin. A chromatin is going to now condense, and that forms what we call a chromosome. We also have what we call a chromatid. A chromatid is simply a chromosome that has not made a copy of itself. So, like the way you can see here, you have got one copy, another copy. That's chromosome. But chromatid is just a chromosome, but it hasn't made a copy of itself. Please, it's different. Chromatin and chromatid, they are two different things. The nucleolus is actually the middle part of the nucleus that is going to be able to form rRNA. Remember, we've got three types of RNA: rRNA, tRNA, mRNA. But the rRNA is formed in the nucleolus. Cells will be able to divide, and depending what kind of cell you have, we have got Lebel cells. What are Lebel cells? These are cells that can easily divide. They have got high proliferative ability. For example, hematopoietic cells, the cells which are found in our blood, which form blood cells, they can easily divide in case you don't have enough blood. Epithelial cells, if you rub off your skin, it will quickly come back. You go and bath, you are rubbing off your skin to quickly come back. Lymphoid cells, B cells and T cells, which are produced to fight infection, these are Lebel cells. Stable cells are also known as quiescent cells. These ones don't normally divide. They only divide if there's an injury. For example, the cells in the liver, we call them hepatocytes. The cells in connective tissue, we call them fibroblasts. And then also smooth muscle cells. We also have permanent cells. These ones don't divide at all. If they die, they are dead, like neurons, cells which make up our nervous system, and then the cells in the heart called cardiomyocytes. So that's why you don't want the heart to be having problems, because immediately the muscle tissues of the cell of the heart begin to die. That is bad. To stop to beat, stop to pump blood, and someone is just waiting to go. Now the cell cycle, these are just stages that the cell undergo to be able to divide. There are only two phases of the cell cycle. I've got interphase and M phase. Interphase, this is preparation before dividing can happen. You have got G1, S, and G2. There's actually also G0. The M phase can either be mitosis or meiosis. So you can see this is the cell cycle here. You have got interphase. You can see it is really a long process made up of prophase, metaphase, anaphase, telophase. But just take note that prophase is the longest phase of interphase. Telophase is the shortest phase. Interphase is the preparatory stage. You have got G1. G is representing growth. So here what is happening is that the cell is making proteins. It's making proteins, and it's just getting to grow like that. Okay. In the S phase, this is S is synthesis. The most important thing is that there is DNA replication. DNA is making a copy of itself. And then G2, there is a further synthesis of proteins and organelles. Anything that wasn't made will now be made. Okay, that is what happens. For those cells which are not supposed to divide, they'll go in what we call G0. meaning that they are resting, they are not growing. And then mitosis is simply the division of cell. When you start with cell, one cell, you're going to end up with two cells. And the one important thing about mitosis is that DNA is going to be maintained. If you started with a cell that has got 46 chromosomes, at the end, the two cells you are going to have will be genetically similar to the previous one, and they're also going to have 46 chromosomes each. For me, I remember this by using IPMAT, interphase, prophase, metaphase, anaphase, telophase, IPMAT, the stages in order. In in interphase, we have already discussed what interphase is. This is where I've got the G1, the S, the G2. Okay. So interphase is a phase of the cell cycle when the cell is doing its normal functions. Normal functions. Normal function. This means that you are not actually dividing the cell or anything. The cell is just doing the normal functions. Prophase. This is where now the nucleolus disappears. The nuclear membrane that covering the nucleus disappears. Okay, so that you get to expose the DNA begins to condense, because it needs to condense so that it becomes visible under a microscope, because it wants to start dividing. And then in metaphase, M for middle, the chromosomes which have condensed now goes on the middle, and they are going to be attached on the centromere by what we call the kinetochore. And then anaphase means A is for away. Those chromosomes which went to the middle of the cell are going to now separate to go away from each other. Telophase means T for two. The cells now divide into two. And then you have got cytokinesis. It's the actual division now of the cells that you formed into two different cells. In a plant cell, we have got what is called a cleavage furrow forming. In an animal cell, a cell plate forms. I didn't include some pictures. Okay, let me use this one. So this was the interphase. Prophase, you can see the chromosomes start to appear, they condense, the nucleolus disappears, the nuclear membrane disappears. Metaphase, the chromosomes arrange on the middle. Anaphased, you can see these guys are attached. They are attached on what we call the centromere. They are going to be separated on this centromere, so we're going to have two different ones. So you can see a red one this side, a red one this side. So metaphase, they are on the middle, attached on the centromere. In anaphase, now they are going to be taken away from the middle. They are going to be separated on the centromere. And then telophase, now they are going to divide, the cells will divide into two. In an animal, that is known as cytokinesis, the division of the cytoplasm. So in an animal cell, that is known as a cleavage furrow first form, like more like a compression is going to happen on the middle here to show where the cell will be able to divide. In a plant cell, we call it the cell plate. While in meiosis, we don't form the same cells. Okay, in meiosis is going to be different. But why do cells undergo cell division? To grow, to repair anything that has been destroyed, or any cells that have been replicated, finished, or to reproduce. These are the functions. You can find an exam, they just mention the functions of the cell cycle. Or why do cells undergo cell division? Those are the ones. Now, meiosis, you start with one cell which is haploid, meaning it has got 46 chromosomes. And then it ends up being diploid, that is 23 chromosomes. Okay. And for such, the actual meiosis is going to be divided into meiosis 1 and meiosis 2. In meiosis 1, you have got the same phases, IPMAT, interphase, metaphase, anaphase, prophase, telophase. Put ones, and then you are going to go to the twos. The only difference is this. In It's the same as the phases for mitosis, but then in prophase 1, there is something that happens that is known as genetic recombination or crossing over, and that is what results in genetic variation in organisms. That is why we don't look like our parents. Why? Because of full genetic recombination. That's why we don't look like your siblings, because each cell that goes to a different individual is actually going to look differently because there was a combination of different chromosomes. And then in metaphase again, the chromosomes align on the middle. But take note, the chromosomes aligning on the middle in metaphase, we actually call them, these are homologous chromosomes. Okay, they are homologous chromosomes. And then in anaphase 1, very important, what you separate in anaphase 1, you don't separate the centromere. What you separate are two homologous chromosomes. You are going to only separate the centromere. You can see, look at meiosis 2. I mean, yeah, meiosis 2 here. This is supposed to be metaphase 2. You can see now you have got the centromere here holding only one cell. Now you are going to separate the centromere in anaphase 2. Centromere is separated in anaphase 2. Anaphase 1, you don't separate the centromere. So that is what happens. You want to be more detailed, prophase 1 has got five other stages: leptotin, zygotin, pachytin, diplotin, dikinesis. The actual crossing over that we are talking about to happen in pachytin, and pachytin is going to be the longest phase of prophase. Okay, so what will be the result of meiosis? We're going to be able to form a cell that is genetically variable from other cells. Crossing over happens, and this is happening in what we call gametogenesis, the formation of gametes. Then you are going to form a primary oocyte. Now let me not talk about this because we're going to talk about gametogenesis in the next class for anatomy. Is there any questions before we go to questions? Is there any question? Okay, let's start. Which of the following is not true about or concerning the mitochondria? They replicate independently from the cell. They are passed on via sperm, contain their own genetic material, vary in morphology between cells. Which one do you think is not true here? Let's go. People, are we in class? Okay, so since people don't want to speak, what to do is I'm going to send these questions, you try them yourselves. If you want to confirm if what you've answered is correct, you can get to inbox me to confirm where you are not clear. So I included a lot of different questions just to make sure that you understand what we've discussed.